Okay, we're ready.
Okay, good. So let's continue, then we can go home. Earlier, I think I have to speed up a little bit, but we can manage that. So I want to give you an idea about something that's important for my research, but maybe also interesting as such, which is that you would like to, for example, find out when you look at a certain response. For example, you compare like an immediate early response to like the ground state. So you always, when you do transcriptomics, you always compare something. It's a case control design, a case control setup. So you have, cells that are in resting state, and then you give them like a growth hormone, or like you take macrophages and you give them lipopolysaccharide, which is kind of this bacterial marker, and they do something. So you have case control setups, and then you see like expression of genes that change. That's what we measure. But then I said that we actually are interested in. So that's a descriptive thing. You see these genes are changing, but often we're a bit stuck, and then we say, okay, now we find these 200 genes that are changing. What do we do now, right? So one way is that we would like to know actually which transcription factors might be the ones that are triggering these responses. And the idea is that if you have like one factor can actually regulate several target genes. It's whole programs of genes that are regulated. But maybe it's only one or two or three regulators, and if they are switched on, then you see a change of some dozens or hundred genes. So we would like to know what is actually the transcription factors, or the binding of transcription factors that is important for a certain response. And one way of doing it is this motive activity response analysis. But for here, I think I only want to show like the concept of that. And one type of data which I like a lot, but it's one way of measuring the transcriptome, and this is done basically by using the capsid that we talked about. So this is a way where you can actually fish out RNA, not by the poly A tail, as we said before, messenger RNA, but we fish out messenger RNA by its capsid. Okay, so we use the capsid to fish it out, and then you do. So this is the capsid, and then you sequence the beginning of the transcript here somehow. So when you sequence only the beginning of the transcript, which is this part, then you would, for example, it could look like this. So you get these sequences, but this 5' end of the sequence is basically the transcription start site, the TSS. And then we are only interested in the very first position of this. And if you then visualize these first positions here, then you can get these kind of histograms. So this is where a specific transcript has started, and the higher the bar, the more transcripts you had starting from that specific position. Okay? So you get these clusters. And actually in reality, they look like this. So this is a genome browser view. I don't know if you have used genome browsers in the course so far. So a genome browser is a tool which is like, you can run it in a web browser. And so you see here a piece of the genome. For example, this is an older slide. This is like human genome version 18, chromosome 12 in certain positions. So this whole window that you see from here to here has a size of 15 kilobases. Yeah? So that's the size you're looking at. 15 kilobases, and it's centered around one gene. And for the purpose here, it doesn't matter what this gene is, but it's the start site actually of this gene, was a TPN6 or whatever. And so when you then use this type of RNA sequencing, which is called cap analysis of gene expression, it's called CAGE, cap analysis of gene expression, then you get, in HeLa cells, you get like a very nice peak here, and in a different cell line, in THP1 cells, you get a peak here. So this is a typical example of alternative transcripts, and up here you have gene models. This is a gene model from UCSC. Have you talked about gene models? No.
Oh, yeah, and gene prediction. Already did that. I don't know.
Gene prediction. So maybe I can 1 minute on gene models because that's important. So we have a lot of different ways of actually defining what a gene is. And one way is, it's kind of the way that the reference sequence database is doing, is that they say there's a lot of evidence on a gene, but we actually have an expert or experts and they are reading the papers on this gene, some papers, and then we define, okay, based on these papers, we can say the gene is like starting here, ending here, has these isoforms, and the gene is important for these kinds of things. So it's done by a manual curation of an expert. So this is the most reliable way you can annotate a gene, okay? And the database which is hosting these genes is called the RefSeq, Reference Sequence Database. It's by NCBI, National Center for Biotechnology Information in the US. And then there is another approach that somebody says, you know, I just take all the information I can get about all the genes, and then I give certain levels of trustworthiness to these genes. So for example, if a gene is coming up in many different databases and it's kind of coming up in some publications and here and there, it's a likely gene that's likely there. But there's other genes where I'm uncertain about, but I still list all of them and I give certain levels of credibility to all of them. So this is the opposite approach to that. And for example, the Ensemble database of the European Bioinformatics Institute is doing that. It's called the Ensemble database, and they have a mix of high quality genes, but also genes where we are very uncertain if they actually do exist. Yeah? So gene models come in different flavors. That's something important, I think, to remember. So here we have, this is the reference gene, ref gene. So this is the very kind of reliable. And here you have 3 gene models for ref gene. And you have one which is starting here. So the box around means the untranslated region. And you have another isoform which is starting here. And very interestingly, you see actually that in HeLa cells, this isoform is used, and in PHP1 cells, that isoform is used. But as I said before, the set of transcription factors here, so you see this whole window is 15 kilobases. So then, I guess, maybe then one of these tick marks should be around like 1 kb or so. So you see from here to here is maybe 2 kilobases. So this window here would be like maybe 1 or 2 kilobases. So you see this promoter region is very different to this promoter region, right? So the way that the same gene is regulated in both cell types or in both cell lines is actually potentially very different to each other. That's very interesting. Yes. But maybe actually for the sake of time I skip these 2 things. So I will not talk about this. Maybe I also skip this one, it's too much. But so maybe you don't really know how I got here now, but basically if you're really interested you can read here. But so these interactions basically tell you that in a certain process, in this case it's actually the differentiation from monocytes to macrophages. Yeah? So this is a cell differentiation process. During that process, you actually have certain transcription factor binding sites which are active during that differentiation. And they regulate each other. But I think maybe we can leave this out a little bit. But you can do networks like that, and sometimes.
Have you used Cytoscape to make this?
Say again?
Have you used Cytoscape or something to make it?
I think this one was probably made with Cytoscape and some add-ons here for the functions. Yes. But then there's now, there's like dozens of methods which draw actual networks. The challenge is how to get the information in about what is regulating what. Okay. Okay. Yeah, but maybe we skip that. So something else that I find very interesting, and I wanted to share this with you because it's kind of actually ongoing research and very hot topic. So I think hopefully I can share my enthusiasm with you. I will make an honest effort at least. So it's basically to elaborate a little bit on the elements which I show here, which are like distant cis-regulatory elements or cis-regulatory modules. And so these areas would be often very small. They are, I would say they are like around 250 base pairs long or something like that. Which is actually a challenge. They are very small, but they are rather important. And like kinetically, you would say that the transcription by itself can work, but it's at such a low rate that basically the gene is not expressed. But within short term, these activators, these enhancers, so enhancers like to enhance the transcription. So these enhancers, if they kick in somehow, they can really boost the expression like by factor of 10 or 100, 1 or 2 order of magnitudes. It's actually very massive. So these enhancers are really kind of the kick-off events for transcription, especially under development. What is tricky is that these enhancers, so these cis-regulatory elements, they can actually not only be upstream, so upstream means like minus direction relative to the gene, but these can actually also be downstream. Okay? So enhancers are equally likely to occur somewhere here or somewhere here, which makes it even more tricky to find them. And they can also actually be in the introns of the same gene. Okay? Most of the enhancers are in an area around, I would say maybe 1 or 2 megabases. So based on here, like 1 or 2 megabases, this direction or that direction around, most of them. But there is actually examples where the enhancers can be 10 megabases apart or whatever. But most of them are around 1 or 2 megabases. So they are close, but it basically means 1 or 2 megabases means that they can easily be in the next gene. So you have a gene, then you have another gene, another one, but the enhancer here is actually regulating this gene. It's not regulating the gene next to it. Yeah? Because three-dimensionally speaking, it doesn't matter, you know, this proximity, they can be very close to each other, even though they're far away on the DNA. And we'll get to that in the end a little bit more. So, yes. And what is very important, which I will come to, is that the enhancers and the promoters have to be in the regions of open chromatin. So, one way of a cell to actually regulate, to silence genes, is by compacting the area. So if you modify the histones in a certain way, then the histones actually fold together with the DNA. So kind of they get very compact. And when the DNA is compacted, then transcription factors cannot enter this compact structure somehow. So they cannot enter. So the way of activating a genome, or for a genome to be active, it has to be open. And if the cell wants to silence the part of the genome, it can basically compact the genome. And this is done by something I'm not talking about, but you have like histone modifiers. So these are like enzymes or proteins that would modify histone state and open and close chromatin. So all of this has to happen in open chromatin regions. That's very important. I think I will only mention this one, that enhancers I have actually so if you think of genes, so genes can be, we say like a gene is specific for a certain purpose, right? But we now know that genes are repurposed a lot. So one gene usually has many different roles depending on the context. And if you look at certain cell types or certain tissues, then there's very few genes that are specific for certain tissues, right? So it is so that mostly genes are expressed in several tissues. yeah, because they have different jobs in these tissues. Enhancers, on the other hand, are rather specific for certain tissues and certain cell types and certain processes. So if you're looking for something that tells you about the identity of a cell or about the status of a cell, then you don't want to look at genes, but you want to look at enhancers. Also, there's many more enhancers than genes. So these enhancers give the cell the specificity. And it's not only one enhancer, but like the composition of how the enhancers is used give the cell its identity. And so enhancers are very important in temporal things, so in development. So for example, I think I don't have a slide on this, but if you look at developmentally important genes, or genes that are important in development, they are basically carpeted with enhancers around them. And when you then look at the high resolution time, then you see it's like these 5 enhancers are active. 20 minutes later, they are off, the other ones are on. Then these are off, the other ones are on. So you have like this kind of really different sets of enhancers that come in rapid succession to regulate. Actually, it's like that, and there's papers. It's like super cool, especially in early development. And this was done a lot in zebrafish, because in zebrafish you can very nicely observe the developing eggs. So that was part of the enthusiasm. So, no, but seriously, like the enhancers have a very important role in development. That's something to keep in mind. And there is some hallmark of enhancers. And so enhancers, they are binding these transcription factors, right? So enhancers have actually transcription factor binding sites, and they are like binding the transcription factors. Do I have this here? I wanted to say something else. So maybe maybe if I would do this slide again, I would say I would focus on that. It's actually interesting when you see an enhancer and you know that this enhancer is regulating this gene, then you find preferentially similar transcription factor binding sites on the enhancer and the gene. So basically, the same set of factors are binding to the enhancer and the gene, even though they are far away. And that helps to stabilize this pre-initiation complex. I guess that's the main message here. So how would you go about finding enhancers? So one way is by doing comparative gene analysis, which maybe was part of the course, like compare different species by genome alignments, for example, a little bit. So when maybe you talked about gene orthologs, so which are like genes that occur in the same gene, basically occurring in different species, so that you would find with alignments, and you can see deviations. You can do that with pairwise or multiple sequence alignments. But basically, it's traditional ways of really kind of testing very specific enhancers. Then there is ways. So we talked about that enhancers have to be in open chromatin regions. So there's actually ways to look at the histones and to assess if the histones are marked for open chromatin or not. So that is because I told you about the chip sequencing on the DNA binding proteins, right? You can actually also do chip sequencing for the histones. So the histone molecules, the histone proteins, they can have several modifications, and depending on the modification of the histone, they are either kind of opening the chromatin or they are closing the chromatin. So, and there are specific antibodies for these histone modifications. So you can actually assess the histones with the chip sequencing. Of course, you can do polymerase II binding, chip assays. Have you heard about, do you know what DNase hypersensitivity could be? It's rather simple. So you have an enzyme, which is called DNase. So the purpose of the enzyme is basically chopping DNA, right? So it's destroying DNA. So if you have DNA which is very compact, then the enzyme cannot attack So the enzyme cannot go in and chop the DNA. But if the DNA is open, it can chop. It's very simplified. So you treat the cells with DNAs, and then you wash away the DNAs, and then you sequence your fragments. So in regions that were closed, you don't have any sequencing fragments. In regions that are open, you have a lot of sequencing fragments. So you can distinguish open chromatin from closed chromatin. Something like that. And if you have regions, then here, if you have regions which have open chromatin, but you don't know what they are doing, there's nothing, so you have specific regions that are open, but there's nothing which you know of, there's no genes or anything. So then you have a certain likelihood that it's actually an enhancer which is sitting there. Yeah? So that's a way of doing it. You also see actually, in RNA sequencing experiments, you can see enhancers which are transcribed. And do I have anything on this? Not really. But maybe I could say that the common way of explaining it is that, so in this model we have the polymerase which is binding here, right? And polymerase is transcribing here. So you find transcript here. So if you imagine that this region is actually super close here, then it actually happens so that polymerase is like wrongly transcribing, not here, but it's actually transcribing this way and this way. At least it's initiating and transcribing a little bit. But then there is a machinery here which actually, which stabilizes the transcription. But this machinery is not present up here because it's not meant to be transcribed. But still you get actually some transcripts close to here and some transcripts close to here. This is a bit of, I don't know, you can call it like an echo of a wrong, or like an echo of this transcription or part of a like wrongly transcribed area. Yeah? And these are basically then the enhancer RNAs which you would see from here. And you can pick them up in RNA sequencing. I will show you how they look like. Okay? So if you know what to look for, so if I tell you that polymerase can also bind here wrongly and transcribe a little bit here and here, then you also already get an idea about how would these enhancer RNAs look like. They would kind of, from a certain point, they would go in both directions, right? But maybe not exactly at that point, but like somewhere there would be a small gap, and then you would see some transcripts here and some here, and they would be less the further you go out. So there would be like a bump of something, and here there would also be a bump of transcripts that would get like lower in expression. And this is actually how it looks like. Maybe we can go directly here. So this is a paper from 2000. Does it say 16, maybe 15? So here you have a gene, EGR1, and this is kind of this cage RNA sequencing signal that I showed you. And there is an enhancer which is regulating this EGR1, and this is an enhancer which has been studied for many years by biologists that are interested in early response. So we know that there is an enhancer. This has been well known before. But when you then actually amplify this area here, or zoom in somehow, then you see a pattern which looks like this. And this is kind of this bi-directional pattern which I talked about. So there's this gap in the middle, and then you have this kind of bump here and the bump here. And this is how like then an enhancer looks like. And if you learn computationally how to find these enhancers, how to find these patterns, and you do that for the whole genome, then you would find something around, we found like 40,000 different enhancers. And when you then look at the tissue specificity of these enhancers, then you can make graphs like that, trees like that, which show you that like fetal tissues have certain enhancers, use like a blood group of enhancers. You have like cerebellum brain, you have like fetal brain, looks a bit different. So you can actually give tissue specificity for these enhancers.
Yes.
So we call this like bidirectional expression. So that's kind of, and what you also see is that when you, so these are very lowly expressed, whereas the gene itself is rather highly expressed. These are lowly expressed. So, what was interesting also is when you look at the dynamics of these enhancers. So this is the same locus, the same gene and the same enhancer, and so here's like we have 30 different time courses of immediate early responses, and here's just some examples, which is like growth factor response of a MCF7 cell line. It doesn't matter so much what the biology is, but here you have like a time course, which is, so this is like the first 2 hours. So this is a 2-hour window here, and then we go to 3 hours, 4 hours, 5. So this is like a bit of a log scale somehow. It's not a linear scale. It's not log either. So this is what happens in the first hour, first 2 hours. So this is like, this we call like immediate early response. And you can look at the expression of the gene here, but you can also look at the expression of this enhancer. And we say here that the expression of the enhancer is somehow a measure of the activity of the enhancer. So you would, and what you see when you look at this, kind of obvious. If you look at the timing of these two things, and here as well, and here as well, and maybe here a little bit if you're very positive thinking.
As long as the enhancer stops contacting the gene, then the promoter also doesn't really work.
Yeah, contacting we don't know, but for some reason the enhancer starts to be active, and then the gene comes directly afterwards, right? So if you look at the like 2 peaks, this is at 15 minutes, this peak, and this peak here is maybe at maybe 1 hour. So the enhancer kicks off at 15 minutes after you kind of add your growth hormone or whatever, and then the gene comes up at 1 hour. So it's in rapid succession. It's like 45 minutes. And here the time window is a bit different. But the point is that for this one gene, we see this pattern that we know the enhancer is regulating the gene, and we see that in the early response, the enhancer is actually coming on just before the gene. Yeah? And in this paper down here, we did this analysis not only for one gene and one enhancer, but for, I think, 150 early response genes and some 100 enhancers over 25 different or 30 different cell types. So we see like a very common, clear pattern that actually enhancers are kind of the kick-off response for this. Very good. So I wanted to briefly talk a little bit about the chromosome structure, and yeah, I don't know, like chromosomes might look actually like this rather than one-dimensional things, of course. And here we are very interested in things that are far away could actually, on the one-dimensional axis, can be close to each other on the three-dimensional. I guess I've said this 10 times now, and I guess you're a bit tired of hearing that. But how could you then, I show you one method, how you can measure that, which is actually pretty simple, but also nice. So if you think of the situation where you have things that are far away, but they are close to each other, so in the three-dimensional space, so they are kind of, say, somehow like on top of each other, like the protein is binding to the DNA, so these two strands of DNA are close to each other like this. So you can cross-link the DNAs, the two strands of DNA here, so the red one and the blue one, and then you would kind of digest the ends off, so you clip the ends off. I leave out all the molecular biology because that's not so relevant. But then you have this complex which is open on both ends, then you ligate one of the ends, and then you remove I don't know, the crosslinker here. And then basically if you open here, then you have one piece here and one piece here. And then we would, I don't know, I would just put this here, put this into a sequencing machine, okay? Maybe that's the easiest. We don't need to care about this boring stuff down here. But we can put this into a sequencing machine. And so you put a linker here and a linker here. And then you sequence these chimera, basically these pieces of DNA which consist of two ends. What do you do with that then? If you sequence these pieces, what can you do with these pieces?
Align it to a reference genome.
Exactly. But will this piece match to the reference genome as it is?
And then the individual parts will match, and then you know which parts are close together.
Exactly. So you have to be a bit clever because also you don't know what the midpoint is, and the midpoint is not always the midpoint. But you have to be, you kind of align by kind of from the ends, and then you kind of align until it fits to two places, right? And you also know that these two places should be on the same chromosome, right? Often, always on the same chromosome. And also, if they are more close to each other, it's more likely. If they're very far away, it's less likely. So you can have some assumptions of how to do that in a clever way. Exactly. And if you do that, you would get something like these contact maps. And so, actually, do I have a? So this. And this is how it typically looks like. So you would have, so this means that, so if something is dark red, it means that this part and this part have a lot of kind of these half-end sequences. And you see that if this is like dark red here, it means that this part here and this part, you have found a lot of reads which had both ends there, basically. And what is also very interesting is that it's very typical to find these kind of pyramids somehow. And is there a, maybe this is even bigger. So before I go into some more details, if we do this in two different species, like in mouse, and these are embryonic stem cells, and somebody did the same thing in human embryonic stem cells, and here we look at gene regions which are kind of syntenic. So when we talk about synteny, we mean that basically these regions are somehow comparable between species. So if you do that in some of these syntenic regions, then you actually see that even the structure of the genome as it looks like here is also kind of preserved. And so if you look at a structure like this, how would you translate this picture into a three-dimensional picture? So if you look at, so this is the way we think, right? This, at least the way I think, somehow. And we have, and we have this picture. So how do you translate this picture into this spaghetti picture? How would you describe the threads on the? Do you know what I'm getting at? Yes.
Maybe each pyramid is like a loop, so it's closer. The elements are closer together. The ones who belong to the same pyramid. And
Yeah, I mean, maybe you saw the next slide already, but no. Anyone else who wants to?
The tips of the pyramids is where it crosses. No?
They are
The tips where it crosses, where the frozen crosses, and then the rest gets slightly further.
Well, so yes, so definitely. So if you at the tip of the pyramid here, for example, you know that this part of the genome was actually crossing this part, right? So that's like any red area means that two things were together and the ends are anchored on these ends of the genome. So I think what I could learn from this is that basically if you have like I should bring like a woolen thread of it, but basically if you have like a lot of them are you have like this thread which is kind of very intermingled here, and then you have like a piece here, and then it's like this, and then you have another one here which is like this, and then you have something like this. But then you have these pieces in between here, which is like here and here and here. So you basically have no interactions from this structure to that structure. But inside of the structure you have a lot of interaction. But not between this and this. So I should put this picture, I will put next time. So I would like this picture here translates or is very nicely explained by a model like this.
So it's fair to say that those pyramids are quantized? It kind of looks that way. Like the
Bins, you mean?
sorry?
Bins are quantized, yeah?
What I mean is like some pyramids are size X and some pyramids are size 2X.
Yes.
I mean based on
Yes,
these
yes,
four. Okay.
yes. It just means that this like substructure is then bigger, right? Double the size of this. Yes. And what is very interesting is that you find a certain type of protein which is called CTCF, which stands for, who knows that? But they are like these, so these CTCF proteins you find exactly in these boundary regions here in between. So they are kind of, it's like a, I don't know if it's a ring, but it's a bit a ring which is kind of holding this together so that you don't have interactions between these structures. And now I should tell you, I should have said this before, these structures are called topologically associated domains. Maybe it was already in the previous slide. Topological domains, topologically associated, it's like TAD, TAD. And maybe you knew that already, but so I think that's a very important point. And now, if we think about, so that is specifically relevant for the enhancer interactions, because, so this window here, how large is this domain? You can see it. 1, 2, 3, 4, 5, 6, 1 million. So this is 1 megabase, right? Between the two tick marks. So say this is 1 megabase in size. So we would basically not expect any enhancer from here to regulate genes in here, right? So if you have a gene here, like one of these, then the enhancers have to be within that tab, basically. So that is something that's important for my research. But what is also maybe important to understand that these tags are actually can be very different between different cell types. So here I show you that they are similar because it's the same cell type in two species, but actually in different species they can be a bit different. I think we don't know. So these experiments, this is called high C. When you sequence, so this experiment is called a high C experiment. Like 3C, 4C, 5C, and high C is all against all with the sequencing. These high C experiments are rather difficult to make, and so we don't have these contact maps for all cell types yet. They will be very important. And I think the last slide I have from this was, another question could also then be why you have a lot of interactions in between. I don't know, there was this model proposed a long time ago, like 11 years ago, that you have kind of these kinds of loop structures. But maybe I just don't look at it. I don't like that so much. So I don't look at that so much. But yeah. So that was my last slide actually. And if I can briefly summarize, and I may do it maybe without the slides, so you listen to me. But so what I was trying to do was, so this whole field of gene regulation has super many aspects. And I think in this like 2-hour lecture, close to 2 hours, I can basically not systematically show everything. It's just super much too much. But instead I chose actually to show you some systematic things in the beginning and then some sneak peeks of some interesting stuff. And basically it shows that there's a lot more interesting stuff actually. But there's also a lot of systematic things. Systematic descriptions are actually rather old. You know, this core promoter, proximal promoter is an old concept. And I mean, the DNA binding has been around for a long time. Now we have this chip sequencing, which is a new method to sequence to do it much better, and that's great. So methods develop, but the concepts are basically very similar. But I also wanted to show you, like, that you can, for example, that the transcription, like the basic model of transcription, that's good to know, what's the basic model, but then also that there's a lot of detail to it. There's this kind of multi-step with the stalling or the pausing and the extension, and that you have, like, you can use then the transcriptome sequencing for finding fusions. That's important for cancer. And that's actually what people are using a lot, actually, that is used a lot to characterize different cancers, to get like signatures for different cancers based on the fusion genes. And then I was spending maybe too much time, but I find this exciting with the enhancers. So to show you that relatively small regions in the genome can have a very important meaning for regulating gene expression. And actually that the reason why we are only finding them now in such amounts is that they are difficult to find actually. So you need a lot of these sequencing technologies to find the enhancers. And then if you have found enhancers, you don't know which gene is regulated by the enhancer because you just cannot see it. So you need these three-dimensional things. You need to have an enhancer, maybe I haven't said that, but you can actually then look into these TATs, and if you have a high resolution sequencing, you can actually say, this enhancer is like very much interacting with this gene here, like in space. So it's actually likely to regulate that one. Plus you can see that if the enhancer is up in activity, then the gene is also up in transcription. So you also, by like a co-regulation, you have also like means to identify these things. So I told you about the enhancer story and a little bit about the chromatin conformation, like the compactness or openness of the chromatin. And I showed you one way of measuring that with this Hi-C protocol, but just more as a concept and skip the details. And yeah, so also that a lot of maybe the methods you have learned in the course, which are, Sam said that some of them are a little bit maybe not in context of what they are used for, but they are like taught a little bit as a method. But actually all of them are useful components for all the work we are doing here. And yeah, I guess with that I would be happy to conclude, and I can stay a few more minutes. If anyone has questions, ask now or later is also fine. Okay, thanks.
So we're very grateful that Carsten came because we don't have anybody that does transcriptomics here at SU, and he's not obligated to come. He's from KI, so we're very thankful.